sophomore. And through the SkoolMentor program, I learned about the identification of putative inhibitors affecting the regulatory regions of selected genes involved in AML. So, what is AML? AML, or acute myeloid leukemia, is a fast progressing disease where myeloid cells rapidly grow, interfering with the production of normal white blood cells, red blood cells, and platelets. The main cause of AML is a genetic change in the DNA sequence of hematopoietic stem cells or myeloid cells. So what exactly happens to the blood during AML? The overproduction of immature myeloid blast cells takes up space in the bone marrow and thus killing the healthy blood cells and platelets. To continue, epigenetics was another major component of treating AML that I needed to understand and learn a little bit more about. Epigenetics is DNA modifications that do not change the DNA sequence and can affect gene activity. We can use epigenetics as a way to hide the gene codes from being translated into proteins, which leads to a possible targeted cure for certain types of diseases. Now that we got into what AML is, the genesis of my work, so we can understand my hypothesis for this research project. My project was to recognize certain genes that cause the manifestation of AML. I researched the gene's direct binding and whether it was upstream or downstream of the target gene. I would then look for inhibitors that could be potentially used to inhibit the activity and in turn prevent AML from further developing. The objective of my research was the identification of regulatory regions associated with certain genes which are important for the manifestation of AML, and the identification of novel inhibitors which could be found on regulatory regions of target genes. My workflow. So I started my project by learning about the terms and concepts related to AML. This included the genesis of my work covered in earlier slides. Then I explored genes related to AML using the cBioportal software and NCBI software. Using the data from these softwares, I focused on a few genes to study for my research. I utilized the cBioportal datasite, which provides different kinds of samples and cohorts for different studies on AML. And based on mutations and most prevalent genes, I selected a few genes for analysis. Then I researched cell lines and pathways that connected the chosen genes together. Lastly, I looked for an inhibitor that could target the genes to limit the proliferation of immature myeloid cells. And lastly, I present my findings. Research project by analyzing cohorts in cBioportal software. Here is a list of some of the most prevalent genes in AML and a pie chart comparing them all together. FLT3, RUNX1, CEBPA are the genes I will be focusing on today. Our gene of interest is RUNX1. The RUNX1 gene provides instructions for making a protein called RUNT-related transcription factor 1. This protein interacts with other proteins called core binding factor beta or CBF beta, which help RUNX1 bind to DNA and prevent it from being broken down. Here is the oncoprint of the gene in the same cohorts. If we were to inhibit FLT3, and then CEBPA in turn would also be inhibited. So finding that CEBPA is actually binding to FLT3 was quite surprising. I found that the binding of FLT3 promoter region was between 1,162 and 1,171 base pairs. I couldn't find the binding sequence of FLT3 to see if there was a direct binding the opposite way, so I decided to search another gene that could directly bind to CEBPA. Again, I went to the KEGG database and found a pathway where AML1 or RUNX1 directly binds to the CEBPA gene. Through this pathway, I looked into RUNX1 and its downstream to CEBPA. This leads me to my results slide. To end off the presentation, CEBPA causes mass proliferation of myeloid cells in the bone marrow. There's no inhibitor that directly binds to CEBPA. Thus, through RUNX1, we can inhibit the expression of CEBPA. My identification of a novel inhibitor included the BET protein, which is still in testing but could be used. To end off my presentation, I'd like to summarize my findings. And so for the discussion, I found direct binding of RUNX1 to CEBPA. I found a cell line which has positive expressions for all three genes. I found one inhibitor known for RUNX1, and in the future, I'd like to test the activity of this inhibitor in the cell line. We can block the activity of RUNX1 using this inhibitor. Thank you.
So the question I have for you is that how frequent is the mutation in the CEBPA in the AML cancer? Like how how frequent is this mutation as compared to the other?
So this mutation is kind of frequent in terms, so there's a 6% mutation. Um, I tried using FLT3 because it has a higher gene mutation rate, but FLT3 seemed to already have many inhibitors being used. So I wanted to find a new research done on new genes. So CEBPA was the gene I chose, and it had a 6%.
Since you're in high school and just started college, it's a great thing.
So, well, the research experience, when I just got into it, I was like really confused. Like I really didn't know what was going on. But like Doc had really helped me throughout the whole process. And then whenever I asked questions, he would always like answer and then give me a full like answer. And it was really fun, like so researching through the different databases, learning about how to like use them and what research and data I can collect from them. That was a really fun experience. And overall, I really enjoyed it.